A pleasant day, cells. This is Group 1 presenting the overview of food and industrial microbiology. Hi, I am the first presenter, Maria Cecil Aringa. Basically, food microbiology focuses on the role of organisms in food, including their role in food spoilage or food production, while the industrial microbiology is an applied branch which focuses on how they can use microbes in a large scale production of economic use. We can see in here that the interest of food and industrial microbiology bloomed in 1800s through the discoveries of Theodore Swan and Louis Pasteur. Figure 1.1 shows the topics of major interest to the food microbiologist, which includes food preservation, foodborne diseases, and quality control, to name a few. The figure shows the various microbes and their associations with food. Note that each group has its own beneficial and harmful effects to food. Generally, each group may contribute to food spoilage and foodborne diseases. Here, these are the important roles of microbes. Probiotics, for example, contains live cells with bacteria that have apparent health benefits to the consumers. In terms of food biopreservation, antimicrobial metabolites of desirable microorganisms are being developed and used in foods in place of preservatives of chemical origin to control pathogenic and spoilage microorganisms in food. Shown in here are the important mold genera, which includes Aspergillus, which is involved in food spoilage. We have the dairy mold, Geotrichum candidum, and Penicillium, which can be used in food production.
Important yeast genera. Yeast are important in food due to their ability to cause spoilage. Many are also used in food bioprocessing. Some are used to produce food additives. There are several important genera. The Saccharomyces is the most important genus and contains heterogeneous groups. Saccharomyces cerevisiae variants are used in baking for leavening of bread and in alcoholic fermentation. Next is Pichia. They are oval to cylindrical cells and form pellicle in beer, wine, and brine to cause spoilage. Species are Pichia membranifasciens, Rhodotorola. They are in pigment red, pink, or yellow, forming yeast and can cause discoloration of foods such as in meat, fish, and sauerkraut. Species are Rhodotorola glutenis. Turolopsis, they have spherical to oval structure. They cause spoilage of milk due to the ability to ferment lactose. Species are Turolopsis spherica, candida. Many spoil foods with high acid, salt, and sugar and form pellicle on the surface of liquids. Some can cause sensitivity in butter and dairy products. Species are Candida lipolytica. Zygosaccharomyces involves spoilage of foods containing high sugar, salt levels. Species are Zygosaccharomyces nosbomeri. These yeasts are termed osmophilic because they can grow in high concentrations of solutes. Important viruses. Viruses are important in food for three reasons. First are some are able to cause enteric disease and thus, if present in a food, can cause foodborne diseases. Second, several other enteric viruses such as poliovirus, ecovirus, and Coxsackie virus have the potential of causing foodborne diseases. Third, some countries where the level of sanitation is not very high, they can contaminate foods and cause diseases. Bacterial viruses or bacteriophages are used in the identification of species or strains by a process called transduction. Bacteriophages can be very important due to their ability to cause fermentation failure. Many lactic acid bacteria used as starter cultures in food fermentation are sensitive to different bacteriophages. These phages can infect and destroy starter culture bacteria, causing product failure. This table shows the human intestinal viruses with high potential as food contaminants. Types of viruses are the picornaviruses, and the examples are the polioviruses, Coxsackie virus A, Coxsackie virus B, ecovirus, and enterovirus. Rheovirus are the examples are the rheovirus and rotavirus. Parvoviruses are human gastrointestinal viruses. Papovaviruses, human Biki and GC viruses. Adenoviruses are human and adenoviruses.
Good day everyone, I'm Michelle Jane Bongabong and I'm going to discuss about the important bacterial genera. Bacterial classification is rapidly changing. The table shows the group, family with the respective genera that currently approved, used, and listed in Bergey's manual. Common bacterial groups in foods. Among the microorganisms found in foods, bacteria constitute a major important group. This is not only because many different species can be present in foods, but it also due to their rapid growth rate, ability to utilize food nutrients, and their ability to grow under a wide range of temperatures, pH, and water activity, as well as to survive under adverse situations such as survival of spores at high temperature. For convenience, bacteria important in foods have been arbitrarily divided into several groups on the basis of similarities in certain characteristics. Some of these groups and their importance in foods are listed in a table.
Normal microflora of some common foods. Pseudomonads, Staphylococci, Micrococci, Enterococci, and coliforms. These microorganisms can be found in the example below: the meat, poultry, and especially eggs. Pseudomonads constitute the major contaminants on the skin of freshly dressed poultry. And also, microorganisms, particularly bacteria and molds, may enter the egg through cracks in the shells or penetrate the shells when the bloom or thin coat of the egg covering the shell deteriorates. So this is that's why some experts advise that it is not safe to use cracked eggs because some microorganisms contaminated the eggs that can cause poison if ever that someone can eat it. It can be poisonous. So fresh meat cut from the chilled carcass has its surface contaminated with organisms. So even though that you you clean your hands and cut the meat, we cannot avoid the contamination from the air, which can be carrier for these microorganisms. So next,
Fruits and vegetables. Infection by bacteria, fungi, and viruses in which spoilage increases. Shellfish and finfish. Shellfish that grow in contaminated water can concentrate viruses and may be the source of hepatitis infection. Milk. The spoilage of pasteurized milk is caused by the growth of heat-resistant streptococci utilizing lactose to produce lactic acid, which depresses the pH to a point where curdling takes place.